Welcome to Grand Rounds, and this is the Kaler Award Day, and our speaker is Daniel Yang, an excellent example of what immigrants to America can provide for our country. Daniel was born in Taiwan and emigrated to the United States and became a naturalized citizen. And he went to Texas, where he attended Texas's finest institutions, UT undergrad, and UT Southwestern for medical school, where he began to excel in research. And really, since then, research has been a critical part of his career. He's done projects both in medical school and residency that resulted in important papers. And this was, I think, capped by last year. He won one of the most prestigious awards for young investigators in cardiovascular disease, which was at the Northwestern Cardiovascular Young Investigators Forum. I have to read it. The Jeremiah Stamler Distinguished Young Investigator Research Award Grand Prize last year. Daniel has had an interest in fellowship in using stem cell technologies to better understand cardiovascular disease, in particular using induced pluripotent stem cells, which he's going to speak about now. And I think what's critical about this talk is some of you have heard this talk a couple of times because Dan's been working on it for a while. This talk gets better and better every time I hear it, in that he takes a talk and he works on it and makes it better and takes feedback from people after the talk, myself included, and has really honed as he adds his more research to the talk. He also makes the entire talk better. And since the Kaler Award is dedicated to the best talk given at research conference, I think it's very appropriate. So here's Daniel.
Okay, good morning. Thank you, April, for the kind remarks. And I'd like to first start off by thanking the organizers and also the Kaler family especially for this opportunity to present my work today. So today I'm going to hope to share some of the preclinical insights that we've made into hypertrophic cardiomyopathy in my new lab and also in my primary mentor Chuck Murray's lab. I have no financial disclosures. So to outline my talk, first I'm going to start off with a clinical manifestation of a novel MYH7 E848G missense mutation and resulted, at least associated with a cardiomyopathy. We think this is an atypical hypertrophic cardiomyopathy variant. Next part of my talk, I'm going to describe how we're using human-induced pluripotent stem cell as a model of this MYH7 E848G associated cardiomyopathy. And then finally, I'll shed some mechanistic insights we've made into the pathogenesis of this systolic cardiomyopathy. And finally, end with some future plans that we have. So a number of years ago at the University of Washington, we had identified a family here with middle-age onset non-ischemic systolic cardiomyopathy. The proband, patient 1A here, he initially presented at age 44 with actually aborted sudden cardiac death. He was actually playing basketball. He had an ejection fraction of 35%, and notably of normal chamber size, and really just some very borderline concentric hypertrophy, kind of maximal wall thickness is 1.0 to 1.1. So nothing that really fit, that was characteristic for dilated cardiomyopathy or really hypertrophic cardiomyopathy. He had a normal coronary angiogram, but despite maximal medical therapy, ultimately expired after a few years from heart failure. His sister presented a few years later, age 60 with heart failure, and she ultimately also passed away. She had an ejection fraction of 31%, normal coronary angiogram, but ultimately passed away from heart failure. The children here, they're early 20s, they're asymptomatic with normal ejection fraction, but they had regional wall motion abnormalities on their echo. So based on this clinical history of sudden cardiac death while playing basketball, concentric borderline mild hypertrophy, and contractile abnormalities, we were a little bit suspicious of a hypertrophic cardiomyopathy, although this, granted, is a bit of an atypical variant. So we decided to call this a familial systolic cardiomyopathy. So the question is, given this family history, is genetic testing indicated? And so per the ACC/AHA 2011 hypertrophic cardiomyopathy guidelines, at least, class 1 performed genetic testing for HCM if for HCM or other genetic causes of unexplained hypertrophy is suspected. So kind of like this case where we have atypical hypertrophic cardiomyopathy, or if another genetic condition could be suspected. It's also reasonable class 2A recommendation to test at least the index patient. We think that identification of a pathogenic variant will be useful for first 3 members to see if they would be at risk for developing similar cardiomyopathy. And once you do genetic testing, is there any prognostic value of this genetic testing? Well, so for the Sarcomeric Human Cardiomyopathy Registry, the SHARE registry, the international database of over 4,000 patients with hypertrophic cardiomyopathy, they followed them for about 5 years or so, and over 60% of these patients underwent genetic testing. And with a composite endpoint here, they found that patients in green with this pathogenic sarcomeric variants, they did significantly worse in terms of their composite endpoints compared to patients who were negative for pathogenic variants in red. The blue line here are sarcomeric variants of unknown significance. And it's not that these buses have only mildly detrimental phenotype, but probably because these are really not well categorized, and they really have unrecognized pathogenic and benign mutations that are kind of lumped in there. But this difference in composite endpoints was mainly driven by heart failure and not so much by ventricular arrhythmias. So yes, doing genetic testing or performing genetic testing can have some prognostic utility. So we ultimately did perform genetic testing on this family with this systolic cardiomyopathy and identified this novel MYH7 E848G missense mutation. And so again, this family members who were affected all had some type of contractile abnormalities, and we found that this E848G allele did segregate with this clinical phenotype. Now, myosin heavy chain 7, as you may recall, is a thick filament of the sarcomere, which is the primary contractile unit in muscle cells. So because of this devastating allele that causes premature cardiac mortality, my primary mentor and I decided to study this family's disease in hopes that ultimately one day we can offer them a precision-based disease-modifying therapy. So there's numerous ways that you can study cardiac diseases. You can study in vivo. So most common is in small rodent models like mice or rats. The problem with that, rodents are physiologically quite different from humans. They have heart rates that are 600 beats per minute, and they express different protein isoforms than from humans. So for example, in rodents, they predominantly express myosin heavy chain 6 and only have very little expression of myosin heavy chain 7. So as you can imagine, it can be very difficult to study this mutation in myosin 7 if the animal model doesn't really express that protein isoform. You can also use large animals. If you don't want to use rodent models, you can use dogs, pigs, and they certainly are physiologically more similar to humans, and they express more similar protein isoforms, so they do have myosin heavy chain 7. The problem is they can be prohibitively expensive, especially for junior investigators starting their own lab. So the alternative is to study an in vitro model. And so in the past, it's always primarily been the primary cells, which are cells that are actually obtained from living tissue. So you could actually obtain living cardiomyocytes from patients. And this is the kind of gold standard with that has an actual mutation and it behaves like the patient. But the problem is it's difficult to acquire these tissue. You have to do repeated cardiac biopsies. And furthermore, once you do obtain these cells and grow it out in culture, they actually don't survive for very long, probably at most about a week or so, and then they die off. Embryonic stem cells is another model that can be used, and they can generate essentially an unlimited supply of unlimited, essentially any cell type, so we can make embryonic stem cell-derived cardiomyocytes. The main issue here, though, is the ethical concerns of how you procure the embryonic stem cells. So finally, in 2007, there's a development of these inducible pluripotent stem cells, and these are stem cells that are made from any terminally differentiated cell, blood, urine, skin, and we can make these any terminally differentiated cell into an iPS cell. And these cells are very similar to embryonic stem cells that they can culture, proliferate indefinitely in a dish, and then they can be differentiated any cell type of interest. So we decided to use patient-specific iPS-derived cardiomyocytes to study this human disease in a dish, given the advantages that I described previously. So we performed skin biopsies from our patients, and we cultured skin fibroblasts, and then we reprogrammed them to these human-induced pluripotent stem cells. From these iPS cells, we're able to, after a series of treatments with cytokines, we're able to differentiate them forward into these iPS-derived cardiomyocytes that look very similar to primary adult cardiomyocytes, at least structurally here. Here we're staining with immunofluorescence for the sarcomeres. You can see the nice characteristic striations longitudinally of the sarcomeres, and this looks very similar to structurally to adult cardiomyocyte. Not only does it look like a cardiomyocyte, but it expresses a lot of those proteins that are similar to cardiomyocytes. So this is flow cytometry, just showing quickly that over 80% of these cells, wild type and mutant cells, express cardiac troponin T, which is specific for heart cells. And then furthermore, these cells actually will beat spontaneously in a dish, as you might be able to see here. This is actually a feature dish, and we're just looking at it with our naked eye. You can see the ripples as they beat. And then when you zoom in under a microscope, you can see waves of these cardiomyocytes just kind of beating spontaneously. And then as we zoom in, you can see it beat very rhythmically. So these iPS-derived cardiomyocytes not only look like adult primary cardiomyocytes, but they express some of the similar proteins, troponin T, and they also can beat spontaneously like their counterparts. So from these iPS-derived cardiomyocytes, we can study the individual cells as they twitch, but we can also grow 1 million of these cells, put them together, and make essentially engineered heart tissue that will look very similar to papillary muscles, and we can study this on a tissue level. Similarly, we could then take more reductionist approach from these individual cardiomyocytes. We can isolate individual myofibrils and study the myofibril twitch kinetics. As you can see here, here's a myofibril that we've isolated with a characteristic striation. So we wanted to see if this iPS-derived cardiomyocyte, this model, can recapitulate the systolic cardiomyopathy that was present in this family. And this project started many, many years ago, back in 2012, when this was very novel. And so we made iPS-derived cardiomyocytes from these 6 individuals highlighted in red, 3 individuals with a clinical phenotype of some type of contractile abnormality, and within the myosin E848G allele segregated with it. And then we also made these iPS cells from 3 individuals without any clinical phenotype and who did not harbor the E848G allele. And so next on this slide, I actually will quickly go over years of work describing how these iPS cells can model this disease. So from the single cells, we can study the individual twitch kinetics. And so here we have 6 different patient lines of iPS-derived cardiomyocytes, and these are average twitch traces from these cells. The 3 blue, or shades of blue, are wild-type patients, and the 3 shades of red-orange are patients with the MYH7 E848G allele with this familial systolic cardiomyopathy. And so that you can see here, the wild-type lines in blue all have about a fractional shortening of about 3%. So every time they contract about 3%. Comparatively, the patients with a familial cardiomyopathy with an E848G allele, they all contracted on about 2%. So similarly, they were able to recapitulate this contractile impairment in these iPS-derived cardiomyocytes. We can study the individual myofibrils, and so when we isolate the myofibrils, and this is a representative twitch force from these patients' iPS-derived cardiomyocytes, we can show that the mutant iPS-derived cardiomyocyte myofibril is actually significantly weaker compared to the wild type. And then furthermore, this also translates on the tissue level. So like I mentioned earlier, we can take 1 million of these cells, grow it together, make engineered heart tissue, and study those tissue. And they're very similar to cardiac papillary muscle preps. We can take these tissue, we can measure the force that they generate, and then stretch it. And every point of stretch, and then remeasure the force. By plotting active force with length, we can generate these Frank-Starling curves. And so here it's quite obvious that this wild-type patient engineered heart tissue at every point of stretch is significantly stronger than the tissue generated from the patient with a myosin E848G mutation. So overall, these findings suggest that these patient-specific iPS-derived cardiac derivatives can recapitulate the clinical phenotype of systolic dysfunction. However, nothing here really demonstrates that this myosin E848G allele is the cause of this disease. Certainly, the E848G allele segregates with the clinical phenotype, but this could just still be an association. So, is myosin E848G previously thought to be a variant of unknown significance? Is this the cause of the systolic cardiomyopathy? Well, so here's a schematic of a normal patient heart cell, and the dogma of biology is that DNA will be transcribed to RNA, translated to protein, and then protein will have different functional abilities. In this patient with the familial cardiomyopathy, we know have one mutation in myosin 7. They have this E848G missense mutation, and we've demonstrated that they will make the mutant protein and it'll go into the sarcomeres. So the question is this a pathogenic variant, this myosin E848G allele? Well, the problem is these are family members, and so they at most only share 50% of their DNA. And so there are other chromosomes, 23 other chromosomes, 10,000 other possible genes that could harbor other mutations that we're not aware of. And any one of those other mutations could potentially actually be the cause of this cardiomyopathy. And this myosin E848G could potentially just be a red herring. It makes sense that it could be, as it is a sarcomeric protein. But to prove that, though, what you really need to do is control for all the other DNA in the so-called isogenic control experiment. And so there's multiple ways to do that. One way is just take a normal wall-type cell, make the mutation, myosin 7 E848G, and study, see if that mutation causes the wall-type cell to become weak. Another possibility is take the patient cell line that's weak, fix the mutation, and see if it improves. Well, long story short, it turns out it's actually easier to break things than to fix things. And so our approach was actually to take a wall-type cell line, and instead of trying to just go in and find that one nucleotide and change it, we actually just decided to break myosin 7. So we knocked out myosin 7. So this one cell line no longer had any myosin 7, and so they no longer have the protein myosin 7. Surprisingly, they'll still have sarcomeres, and these cells will actually still beat. And that's because these young iPS-derived cardiomyocytes actually express this different isoform myosin 6, and so they're able to still form sarcomeres. So finally, with this kind of background, with this isogenic background, this myosin 7 line, knockout line, we can interrogate this myosin E848G allele. And so I took this line, I would then rescue it with adenoviruses carrying full-length myosin 7 heavy chain 7 wild type, or I will rescue it with adenoviruses carrying myosin 7 with just a single E848G mutation. And this way, because it all came from the same cell line in this isogenic background, the DNA, the rest of the chromosome, is all identical. And the only difference is this adenovirus with one single point mutation carrying the E848G allele. This way, we can more confidently attribute whatever findings we have to this E848G allele. So this is what we found. So here we're looking at single cells, iPS-derived cardiomyocytes, and we're using our isogenic line, this myosin 7 knockout cardiomyocytes. And here's just immunofluorescence showing a bunch of these iPS-derived cardiomyocytes. We zoom in here on one cell here, and white is alpha-actinin, which stains the Z line of the sarcomere, so you can see the characteristic striations right here. So they're sarcomeres, even though you don't have myosin 7 because of myosin 6 isoform is still present. But notably, you do not have myosin heavy chain 7. So this knockout line truly does not have myosin 7. And so I can also take this line, instead of treating it with just this green fluorescent protein, this virus with a control virus, we can treat it with the myosin 7 full-length wall-type virus. And here you also again see nice characteristic striations of sarcomeres. And then now you also see red. So you have myosin heavy chain 7 because we're giving it through this adenovirus, and you see the nice striations. Similarly, if we give myosin 7 with the E848G mutation, you'll see striations both in white and red, demonstrating that this E848G allele does in fact go, this protein will go incorporate into sarcomeres and potentially act as a poison peptide. The key experiment is functionally, what does this E848G allele do? And so once it goes into the sarcomeres, what we found is that in these myosin 7 knockout isogenic line, compared to control cells transduced, which is this green fluorescent protein, or wild-type cells transduced with full-length wild-type myosin 7, the cells transduced with MYH7 E848G are significantly weaker. They have a fraction shortening of 2% compared to about 3% in the control cell lines. So this finally strongly suggests that this MYH7 E848G allele is the cause for this patient's systolic cardiomyopathy. And so previously labeled a variant unknown significance, this data would suggest that it is probably a pathogenic allele. Now that we've confirmed that this allele is most likely a pathogenic allele, how exactly is this mutation causing a systolic cardiomyopathy? And so if we hope to develop a novel disease-modifying therapy, we would like to know how it's broken before we can attempt to fix it. And so right around this time, shortly after the publication of that paper, we actually reclassified this from a familial systolic cardiomyopathy to better appropriately called it a hypertrophic cardiomyopathy. And that was because there was a presentation of a new patient that was previously unaware to this family or to the healthcare providers. This patient presented about a year ago to University of Washington the first time, right here. He's 57 years old. He also presented with heart failure. He also had ejection fraction of 31% by MRI with normal chamber sizes, so it wasn't consistent with dilated cardiomyopathy. But this time, this patient actually had severe asymmetrical septal hypertrophy, more classic for hypertrophic cardiomyopathy, and he had septal wall thickness of up to 1.9 centimeters. He was a carrier of the MYH7 E848G allele also, and his course was quite rocky. He required actually a total artificial heart followed by a heart transplantation. But this quick table here summarizes the clinical manifestations of this family in patients 1A, 1C, and 1D. And so what we found is that these patients were presenting somewhat early in their 40s to very early 60s. They all had systolic dysfunction, pretty significant systolic dysfunction, with ejection fraction in their 30s. But notably, these patients all at least had borderline to severe concentric hypertrophy. So we think this is a hypertrophic cardiomyopathy variant, albeit a little bit atypical, as their primary dysfunction appears to be systolic dysfunction. So kind of going back to my original question, how does MYH7 E848G mutation cause hypertrophic cardiomyopathy? We know that this mutation will go into sarcomeres, as demonstrated previously in the isogenic line, but somehow it's still a black box how it results in ejection fraction of 35%. So from this recent patient who underwent a total artificial heart, we actually got a lot of data. So we actually were able to acquire his total heart. We sampled a couple places in his heart, and we measured the protein levels of myosin 7. So here is an immunoblot measuring total myosin 7 levels. Here's myosin 7, here's troponin I, here's alpha sarcomeric actinin. These are cardiac specific proteins, so it indicates that all our protein samples have cardiomyocytes. And GAPDH is just a loading control, demonstrating that we loaded the same amount of protein per well. We have two samples of non-failing human left ventricle samples, and these are actually from two different individuals. And then these next 6 samples are all from the same individual who underwent a total artificial heart who has this E848G allele. And we're here sampling 2 kind of random places in the septum, and then 4 other random places in the left ventricle. And what's striking that we found was that there was quite a few areas in his heart where there is very little, almost non-detectable myosin heavy chain 7. That was surprising. And so going back to our schematic, what this means is that in this hypertrophic cardiomyopathy, it seems like they have at least decreased levels of total myosin heavy chain 7. Well, so how does myosin 7 E848G lead to this reduced total myosin 7 protein levels? A couple hypotheses. It could be due to decreased protein synthesis, or it could be due to increased degradation. And degradation is generally through this common proteasomal pathway. So right now, we don't know which one is the primary driver of this loss of myosin 7, but we at least can determine this experimentally. And so what we did quantitative PCR to assess for the total myosin 7 heavy transcripts in this non-failing human heart and compared to one of our non-failing human controls. and we found really no significant difference in the mRNA transcript levels, suggesting that in this hypertrophic cardiomyopathy cell, the problem is not protein synthesis. They have approximately normal amounts of myosin 7 mRNA, and so they should be making myosin 7, but for unclear reasons, they have a decreased level of myosin 7. So our focus turned to possible degradative pathways. And so what we decided to use was go back to our isogenic line, where we knocked out myosin 7 and just introduced a wild type of mutant myosin 7. This way we can more confidently reassure ourselves whatever our findings are is truly due to this one point mutation. And so here, in this mutant blot, I'll kind of walk you through. Similar scheme, we have myosin 7, we have alpha-sarcomeric actin, and we have a loading control, GAPDH. These are myosin 7 knockout cardiomyocytes, so our isogenic line. MOI is multiplicity of infection. It just means number of viral particles per cell. So if you have this myosin 7 knockout line, if you don't add any virus, well, not surprisingly, you don't have myosin 7. As you start to add more myosin 7 virus, you titrate 20 copies, 80 copies of the virus, you have a dose-dependent increase in total myosin 7 protein levels. Makes sense. We did the same thing for the mutant virus, the virus carrying the E848G allele, and as we titrated in dose, there was a dose-dependent increase in the myosin 7 levels. However, it's kind of interesting that despite we were careful to titrate the same amount of viral particles, there was overall less protein here in the mutants compared to the wild type. Well, what we were interested in is in what happens if you block the proteasome. So we were suspicious that there was a degradative process that was going on. And so we found that the wild type cell, if you block the proteasome, this MG132 compound, you'll get a slight increase in myosin 7 total protein levels. But in the mutant cell line, if you block the proteasome, you get a dramatic increase in the mutant myosin 7. And so that's shown here in this Western blot. If we look just at the 20 MOI dose of the wild type myosin 7, if you block the proteasome, it probably doubles in intensity. And so that's quantified here as in myosin 7 turnover. If you look at the 80 MOI dose, it's about the same. And so that's quantified here. However, the mutant myosin, in the 20 MOI, there's not much, you can barely see a band, but that band is clearly evident now once you block the degradation and you get accumulation of myosin 7. And that's quantified here. There's a very high upregulation, accumulation of this mutant myosin. And similarly, even at a higher dose, 80 MOI, there's a significant accumulation of myosin 7. So what we found here is that there is an increased proteasomal degradation of this myosin E848G in these cardiomyopathic cells compared to the wild type. So the next question is, well, what mediates this increased proteasomal degradation of myosin 7? Well, so it's known that myosin 7, this is already previously established in the literature, it's known that myosin 7 will get targeted by MRF1, the muscle ring finger protein 1. MRF1 is an E3 ubiquitin ligase. It will target specifically myosin 7. It will ubiquinate it, and this polyubiquination of protein is what causes proteins to then be shuttled to the proteasome to be targeted for degradation and then broken down. So given this background, we asked, does MRF1 play a role in the pathogenesis of myosin 7 E848G cardiomyopathy? And so going back to our isogenic control line again, so that we can confirm that our findings are due to this mutation, here is MRF1 in our schematic. MRF1 will mediate the ubiquination of myosin 7, shuttle it to the proteasome, cause it to be degraded. This is again is a immunoblot, similar scheme of the myosin 7 knockout cells. We add in wild type myosin 7 with just some control siRNA, so it's not targeting anything. We get expression of myosin 7, we get basal levels of MRF1. If we block MRF1 with the siRNA, so that will target MRF1 transcripts, that will reduce MRF1 protein levels, and we found a slight increase in myosin 7 accumulation. So that's demonstrated here in this Western blot. These are wild type myosin 7 viruses. If we target with MRF1 siRNA, we can knock down very significantly MRF1 protein levels. We find a very mild increase in myosin 7 protein levels, and that's quantified here. So nothing statistically significant. So the baselines, there might be a little bit MRF1 mediated degradation of myosin 7. But what we were interested in is what's going on in the hypertrophic cardiomyopathy cells. And so in the mutant cell line, we can transduce them with this adenovirus carrying that E848G allele. And we get approximately equal amounts of protein here. We can titrate in different amounts of viruses. What we found is that if we inhibit MRF1 with the same siRNA, we will knock down MRF1 protein levels with this MRF1 siRNA, but then this time we get more of a significant upregulation or accumulation of this MYH7 E848G protein, and that's quantified here. So this suggests that there is that MRF1 accelerates the proteasomal degradation of MYH7 E848G. So this is our current working hypothesis for MYH7 E848G hypertrophic cardiomyopathy. If you have a cell with 2 normal copies of myosin 7, you have normal sarcomeres and you have a normal functioning heart. If you have a different cell with a mutation in myosin 7, and specifically E848G allele, we have data that suggests that there's a MRF1 mediated degradation of this allele, and somehow that results in a broken heart, presumably due to insufficient amount of potentially myosin 7. What we've also found is that if you can block MRF1 with an inhibitor like this siRNA, you can reduce the amount of myosin degradation and thereby increase the amount of total myosin 7 and hopefully cause the improvement in these cells. This is right now still unclear. We don't know what functionally will happen when we block MRF1, but this is of intense ongoing experimentation right now. We actually may have the results of this probably within a week or two, so if you're dying to know, you can reach out to me in a month or so. So if this actually works, this would be great, obviously, for this family, and this essentially will be potentially life-altering for them. But the question is, would this MRF1 inhibitor also be more generalizable? Would it potentially work for other mutations in myosin 7? And so this kind of leads me to the other part of my talk. So kind of shifting gears slightly. We had another kind of interesting observation. So this is a protein sequence of myosin 7 right around that 848 locus. This this glutamate E is conserved from mice to humans. As MYH7 E848G converts this E to glutamate. It turns out, so this is the schematic of myosin 7. In blue is myosin 7, in green is MYH7 binding protein C. It turns out our mutation is right here in the so-called S2 neck region of myosin 7. And our mutation right here is thought to potentially cause a disruption of this interaction between myosin 7 and myosin binding protein C. When we look at myosin 7 a little more closely, here is myosin 7, all the amino acids from 0 to 1900. If we look at all the known human pathogenic mutations in myosin 7, we found that there's a peak here, right where the S2 neck region is, and that's right where our mutation is. So a lot of the mutations in myosin 7 are right here. In fact, it's the second most commonly mutated spot in myosin 7 that causes human disease. If you zoom in a little bit more closely at this S2 fragment of myosin 7, so around 800 amino acid locus, our mutation is right here in the middle, E848, and we mutate this to a G. This is surrounded by a bunch of these small triangles, and these small triangles are known human pathogenic disease, patients with hypertrophic cardiomyopathy and dilated cardiomyopathy. So this led us to make another hypothesis that potentially all this clustering of mutations, pathogenic mutations, potentially what they're doing is they're disrupting the normal function of this protein at this area, which is to bind myosin binding protein C. And so our hypothesis is that mutations that impair myosin 7 and myosin binding protein C interaction will lead to this contractile dysfunction that we saw in this family. And so to test this, we turned to a different assay. We used this yeast-2 hybrid assay. And what we've done here, we've set up the experiment, and I won't go into too much of the details, but we set up the experiment such that if myosin 7 and myosin binding protein C interact, yeast will grow. That's just how the experiment is set up. If we make this simple point mutation, MYH7 E848G, if they don't interact, the yeast don't grow, and that's what we found here. This white fluffy is the yeast growth. And so this suggests that if we don't see yeast growth, that there's no longer any, that this E848G mutation disrupts this protein-protein interaction. What was interesting, we had a collaborator who was also kind of studying hypertrophic cardiomyopathy, but they were interested in myosin binding protein C, and specifically in this E258K mutation that also seems to cause systolic cardiomyopathy. And so they found in this very similar yeast-2 hybrid assay that if they made their mutation in myosin binding protein C, which is called hypertrophic cardiomyopathy, and they put in wild-type MYH7, they no longer see yeast growth. So suggesting that this E258K mutation in myosin binding protein C can also disrupt protein-protein interaction. Well, why do we care? What we found, very interesting downstream, so this is our work here, we found that in our engineered heart tissue, patients with this familial cardiomyopathy, the E848G mutation, their maximal twitch force in engineered heart tissue was substantially weaker compared to wild type. Similarly, our collaborators who are studying the myosin binding protein C mutation, they found that in their engineered heart tissue with that E258K mutation, that they were also substantially weaker compared to wild-type myosin binding protein C. This led us to wonder that mutations that impair myosin 7 or myosin binding protein C protein interaction, they potentially will lead down this common pathway of systolic dysfunction. And so where am I going to go with this? So future direction, we're currently working on developing a very high throughput assay to make all possible mutations in this MYH7 S2 region. And then we're going to perform thousands of yeast-2 hybrids, but in bulk culture. So everything will be growing at 1, 1, 2. We're then going to rely on the yeast to empirically test all these mutations for us to determine the effect of these individual mutations on protein-protein interaction. And thereby then correlate this with known human data to see if low interaction, as predicted by yeast, results in a human cardiomyopathy, and if preserved interaction, as predicted by yeast, results in benign or asymptomatic patients. I then hope to extrapolate this data so that geneticists, when they're seeing patients in clinic, they can then use this data to then predict novel mutations that are presented in clinic, whether they will be detrimental or not. So from this experiment, if we're successful, we hope to generate what's called a heat map here. That will essentially be a lookup table. I'll walk you through this. Here we have kind of representative area of the MYH7 S2 sequence. And so here's the sequence from 0 to 70. Here are all the other possible mutations that we can make from every single locus. We can, since there's 20 amino acids, we can make essentially 19 other possible mutations. The yeast will tell us each individual mutation what the strength of the protein-protein interaction is. And so if it's white, it'll be color-coded. It will indicate that it's as strong as wild type, so it's normal protein interaction. And we know wild type has normal heart. What we'll determine is that if yeast predict a weak interaction, blue, we'll see if that correlates with human cardiomyopathy. And then the yeast could potentially also predict stronger interaction, and that will be determined here, shades of red. And we'll see if that results in also an abnormal phenotype. So let me kind of walk you through here. If a patient presents in clinic, and if they say, for instance, had a mutation here, if they had like this one locus N16, if they mutate that to, like, say, E, and that is predicted by the yeast to be white, then you would predict, well, at least the yeast have empirically demonstrated that this protein function is still preserved despite this mutation. And then potentially this will translate into normal heart function. And this is important for patients too. And you can provide them reassurance, and then they don't have to worry. Because patients, essentially, if they are labeled as variants of unknown significance, that's not a benign finding. They have to live the rest of their life with this, and essentially they're in what's so-called a genetic purgatory. They just don't know if this variance is significant or not. On the contrary, if we had a different patient in clinic, if they had a mutation here, like say V11, and it's mutated to, let's say, a R, well, now the yeast is predicting it's kind of bluish purple, and that would suggest worse protein function and presumably potentially worse heart function. So broken heart. We currently, unfortunately, don't have any disease-modifying therapy to offer these patients, but at least this is prognosticated based on the prior SHARE registry that I presented earlier. But if the prior part of the project, the MRF1 inhibitor, if that works, it would be lovely if we can treat them with MRF1 early in life and potentially fix their hearts and potentially alter the natural history of their mutation. So with that, I'd like to conclude. I hope that I've successfully introduced the idea that human-induced pluripotent stem cells can model human diseases in a dish. I've given an example of this patient-specific and isogenic iPS-derived cardiomyocytes, how I can recapitulate the clinical phenotype on a cellular level, myofibrillar level, and also on a tissue level. And we've confirmed that this MYH7 E848G allele is a pathogenic HCM mutation. We have some mechanistic data that suggests that MRF1 depletes total MYH7 protein level by accelerating the degradation of myosin 7 in these cardiomyopathic cells, and then potentially contributing to the cardiomyopathy. And then we have efforts that are underway to determine if restoring normal MYH7 protein levels is beneficial. And then finally, we've shown that mutations that disrupt normal myosin 7 and myosin binding protein interaction is associated with contractile impairment. And we're currently developing high throughput assays to see if the loss of this protein-protein interaction can be used to predict pathogenicity of various new mutations. And so with that, I'd like to thank my new lab members, Guy Everett and Megan. They did the bulk of this work. I'd like to also thank my primary mentor, Chuck Murray, right here. Without any of this, wouldn't have been possible. And these are many individuals in the lab who have contributed directly to this work. And this is a picture of our lab. I'd like to also thank April's lab for contributing the non-killing human heart samples, and then many other collaborators here, Mike Rainier's lab, University of Washington, Doug Fowler's lab in Genome Sciences, and our collaborators in Wisconsin. I'd also like to especially thank the Kaler family for this opportunity to share my work here today, and also for the Bruce Laughlin, Block, and Dolson support from the Division of Cardiology, Ice Cream, and the VA. So with that, I'm happy to take any questions that you may have.
Lovely. So I'd like to present Daniel with my questions to be Grace Marie Kaler and Grace Kaler was an employee in the University of Washington, worked in multiple research labs. It was known as that person you go to when something doesn't work, who helps teach you how to make it work. And so I think it's great that her legacy here in promoting investigation and helping us promote science in the Division of Cardiology. Thank you, Daniel.
That's a good question. So for the audience in Zoom, the question is, should we rename these diseases? And kind of what Dr. Semper-Terrell is alluding to, it's a bit of a spectrum. Hypertrophic cardiomyopathy could be on one side spectrum, dilated cardiomyopathy is on the other side spectrum. What our work has kind of shown, at least with this one example, is that it may not always be so clear cut. It may not just be simply how the heart looks physically. It may not just be hypertrophied or dilated. We have some patients that are hypertrophied and then they yet still have contractile dysfunction, but not dilated. I think what the SHARE registry has demonstrated, and I think that's useful, is that pathogenic sarcomere mutations, that is prognostic, and they will have adverse outcomes. And if we can somehow intervene early in life, we could potentially alter the disease course. So I think it may be useful in the future to label patients with pathogenic sarcomere mutations versus patients without, with such a benign mutation, and that might be useful in the future. Yeah, I don't know. Are they going to be superhuman? That's a good question. So we'll have to see what the yeast predict, and then we'll see if we can identify those patients and see what their clinical phenotype are. Maybe they'll have increased hyperdynamic function, or maybe having too strong of interaction. Maybe if you can't have normal dissociation between myosin and myosin fibers, maybe that will also be something maladaptive, and maybe they will have a distinct clinical syndrome that we're not aware of. But this experiment may help.
you could have an impairment of the interaction or you could have a decrease in the total of one protein. So is the way to screen to look at the potential for protein interaction or the susceptibility to accelerated chromosomal degradation?
Yeah. Absolutely. Good question. So let me try to summarize this from Dr. O'Brien. He's pointed out, so I've mentioned two types of mechanism. One mechanism is purely on just this loss of this protein interaction between MYH7 iPSC, and this is just localized to the S2 domain. Does that predict all potential mutations or pathogenic mutations, or can MYRF1 potentially identify other mutations, and essentially would it be could it be outside this S2 region? And so I think the answer is quite possible, and we were actually trying to set up. I didn't quite go into details about this other experiment, but we are going to look into that to see if MYRF1, the variants that MYRF1 identifies that are being accumulated or that is targeting, to see if those are pathogenic. So kind of very broadly, what we are currently making is we're making another isogenic line of MYH7 that we can easily introduce any potential variants. Okay, and so this is very kind of cutting edge genetic engineering. So the full MYH7, we can engineer any type of mutation. And what I'm trying to do is then knock out the decreased MYRF1 and see which variants accumulate. And so if we're smart about it, if we tag, let's say, MYH7 with like a green fluorescent protein, and then if we identify how much green fluorescent protein accumulates, potentially MYRF1 can help identify pathogenic variants for us. So that is actually 2.0 that we're working on too. Not quite there yet, but it's on the back burner.
I think you said that the patient who started all this with that it presented with a cardiac arrest. What's the like?
What's the mechanism? Yeah, I'm not sure if I have that slide, backup slide. So what we've done, we have some just very preliminary data that we looked into the calcium transients, and we didn't really see any obvious difference in their calcium transient. So we haven't pursued that as much as the possible arrhythmias in this family. We do know they have quite a bit of fibrosis. The last patient came in, he did a cardiac MRI. There's a lot of patchy fibrosis, but at least in our IPS model, the lowest hanging fruit was contractile impairment. This was readily evident, and so we kind of pursued that. And yes, that's a whole other field that we haven't really looked into, is that arrhythmias. But out of the 3 patients that have very advanced disease, only one of them actually had any significant ventricular arrhythmias. And so far, actually, the children, they actually have preemptive ICDs, and they so far haven't had any appropriate therapies yet. So that is unclear if this mutation will cause arrhythmias, or if the arrhythmia in that initial index patient, if that was just due to his subsequent end-stage heart failure. Right. Right. Yeah, good question. So Dr. Otto is asking, since this is a heterozygous mutation, so there's one normal copy of MYH7, one mutant MYH7, why is like the force that I've shown in the engineered heart tissue, why is it like 80% decrease when the heart is not reduced by 80% functions, might be half? And the short answer is we don't know. There must be other neurohormonal, other pathways that are activated to kind of preserve or at least improve heart function that we're not fully recapitulating in these in our IPS disease and fish models. There's probably a bunch of catecholamines that are present in the circulatory system that's trying to help improve the function of the heart. Because yes, if your heart function was 80% normal, you'll probably quickly pass away, but there are at least adaptive functions that must be present, but then eventually over a 4th, 5th decade of life, then they'll run to premature cardiac problems.
I got 2 questions. Hopefully the first one's an easy one.
Okay.
With the issue about why do these patients develop hypertrophy?
And in your iPS cells, I forget, were those mutated iPS cells hypertrophied, or were they similar cell size to wild type?
Yeah, good question. So Dr. McClellan is asking if we're going to label this a hypertrophic cardiomyopathy phenotype. Did the initial part of the project, did those patient-specific iPS cells, they hypertrophy? I didn't show the data, but the answer is no, they actually did not hypertrophy, which is actually not terribly surprising because the actual patients only had very borderline left ventricular hypertrophy. They were 1.0, 1.1. This latest patient did present with clear-cut hypertrophy with a septum of 1.9 centimeters. We haven't yet made that patient's iPS cells to see if his iPS cells will hypertrophy. So why will, if we think this E848G allele is the cause, why is it causing hypertrophy, significant hypertrophy in some patients and not so much in others? And the answer is that I don't know. There's probably other environmental influences or potentially other secondary hits in that particular patient that allowed him to hypertrophy and not so much the rest of the family.
Then the second question is, you have postulated that these mutations lead to a disruption of the MYH7 interaction, and that leads to the cardiomyopathy. But my question is, why you've shown that this mutated protein activates the proteasomal degradation pathway. The question is, why is this point mutation doing that, as opposed to other sort of point mutations? Have you taken some of these mutations, ones that you're predicting block the interactions, put them into protein modeling, sort of models to see is the reason why that is it really the interaction, or is it that these proteins get so misfolded that they activate that?
Yeah, good question. So the answer is that we have not yet done that, but we want to do that. That's a very good question. So yes, so the goal is to make more adenoviruses, or potentially this other cell line that we're currently in the works of doing, is to make other mutations. We can study if they're in the S2 domain, study if they see if they will disrupt protein-protein interaction, but then also see if MRF1 is causing upregulation of or accumulation of this degradation of this allele. So we haven't yet done that. We can do that very easily just by making more adenoviruses, but we're still trying to make actually a more kind of elegant genetic line to test that. Okay, sure, yeah.
First description rather than a phenotype first description, right? In this case, it would be difficult because it started out as a very uncertain. You don't want to prejudice it.
Correct.
For all the elegant that you have. My question is in your pedigree. So individual 1A is connected to individual 1D, who you ascertained later, by two people who would be opposite characters.
Okay.
And so I wondered, had that labeled as question marks in your pedigree? And of course, we now know that they must be American. I'm wondering if you know anything about their phenotypes.
Let's try to find that. So which patients?
So you added them one.
Oh, right.
Then that proves that.
Right. Yep. Correct. Yes. So I do not actually have the clinical data of that family, of that generation of family members. So you're asking about these question marks, right? Yeah. So unfortunately, I don't have those. We might be able to. We probably can reach out to this individual and ask them.
Yeah.
Yeah. Yeah, we can go back.
Yeah, so we could contact them.
But yeah, it's interesting. And with regard to environmental, he was
All right.
this is maybe that. No, think about
I told her that's years of work.
Yep.
You know, I take care of it for some of the family members. And what's amazing about that is they have normal systolic function. They have marching by itself. Very little chronic velocity non-existent. But that's sort of speaks to sort of the genetic experience over here. Variability that occurred not only with this specific genetic change, but the genetic change that's many cardiomyopathy. I don't know if that has to do with like the gene interactions that you're talking about with you see what has to do with the degradation of the function structure. But just highlight the fact that having this even a genetic variant, but we always equate with and that is a mystery to me by that.
Yeah.
That's more like this.
Yeah, that's a good point by Dr. Owens, is that the penetrance of hypertrophic cardiomyopathy is certainly not 100%, and you can have patients with the same pathogenic allele that's demonstrated here, some with overt hypertrophy, close to 2 centimeter ventricles, while others will have just very minimal hypertrophy. Others may actually go through the rest of their life fine, and it's not exactly clear to us why, but there must be other environmental factors.
Thank you.
Okay, thank you.